In this module, we will discuss the tissue-specific functional profiling, how it is done, and what are its advantages, and what were the different experimentation which was performed to carry out the tissue-specific functional profiling. Now, one of the approach to genomic functional profiling is to observe the tissue specificity of the genes. You know, in the tissues, there are specific genes which impart specificity to that tissues, and those genes, they can be inactivated by the mutation. And what are the approaches to study? Now, in that case, Lee and Lim and their colleague, they actually used two microRNAs to knock down the expression of genes in human HeLa cells in the culture in laboratory. They took HeLa cells in the laboratory, cultured them, and knocked down the expression of genes. Now then, they looked at the profile of genes whose expression was significantly reduced. What they found? They found that the gene's expression was significantly reduced, and they marked those genes whose expression was significantly reduced in the HeLa cells. And they were MIR125, 1 to 4, that is the microRNA expressed in brain. Knock down the expression of genes that are expressed at low levels in the brain, MIR1 to 4. Similarly, MIR1, that was the microRNA which was expressed in muscles, and it knocked down the expression of genes that are expressed at low level in muscles. So MIR was, as a matter of fact, knocked down expression of genes in the muscle. The two miRNAs, they shifted the expression of genes in HeLa cells toward that seen in the tissue in which the respective iRNA are prominent. So what happened that these two miRNAs, they shifted the expression of genes in HeLa cell toward the tissues where RNAs, they were prominent. Now, again, Lim and colleague, they found, or they introduced the double-stranded miRNAs into HeLa cells, and then used microarrays to measure the level of mRNAs purified from the treated cells. So what they did, they introduced the double-stranded miRNA into the HeLa cells. And the result was, it was clear reduction in the concentration of 100 or more RNAs with each microRNA. What happened? That there was reduction in the concentration of 100 or more mRNAs with each miRNA. So each miRNA, it reduces the mRNA's concentration 100 or less. The expression level of 10,000 human gene in each of the 46 tissues, that was plotted by Lim and their colleagues in the same way. And the tissue-specific expression profiling, that could be done by examining the spectrum of mRNAs whose levels are decreased by an exogenous miRNA. So those mRNA whose level was decreased by exogenous miRNA, that was detected and examined and comparing that to the spectrum of expression of genes at the mRNA level in various tissues, they were able to specify the specific functional profiling of the tissues, or they could predict that how the specific functional profiling of the tissue could be studied or could be done.